Hello, my name is Amanda Bowman, and welcome to my talk on identifying target sites for placental therapeutics through the comparison of normal-term pregnancies and intrauterine growth-restricted proteomes. While that may sound complex, I'd like to break it down by showing you a series of 3 pictures and asking you to find the commonality between them. Here we have Beyoncé, Kim Kardashian, and my grandmother. Now, besides all of them being stunningly beautiful and famous, the commonality I wanted to focus on was that they all have experienced some form of pregnancy complication within their lifetime. Complications that have been an issue since my grandmother was having babies. Complications that affect Black women at much higher rates than any other racial group within the US. And complications that can occur even if you have as much money as Kim Kardashian. The pregnancy complication I would like to discuss with you today is called intrauterine growth restriction, or IUGR for short. This is where the fetus's birth weight is lower than the 10th percentile for their gestational age. Different elements contribute to their slow growth rate, including maternal, fetal, and placental factors. My lab conducts research on the placental insufficiencies that can cause this IUGR, which are still largely misunderstood. Recent analyses of omics data, which looks at protein, transcriptomes, and other biological molecules, have advanced placental research to explore pathology such as IUGR at a fundamental level. IUGR omics suggest altered gene expression and/or genetic mutation could possibly contribute to the condition. The purpose of my study was to compare the placental membrane proteomics of normal-term pregnancies to establish placental sufficiency under optimal conditions, then to identify publicly available IUGR omics data, including proteomics and transcriptomes, for comparison. Our work is novel in that we are directly observing proteins found in the syncytio-trophoblast microvillus membrane of the human placenta. We hypothesize that placentas from IUGR cases will have distinct and/or mutated protein and gene expression from that found in normal-term pregnancies. Isolation of the syncytio-trophoblast membrane was performed on placentas from term deliveries with no reported complications. From these membranes, resulting peptides were recovered and analyzed at UC's proteomics core, where we matched our peptides to a database that identified what genes were present on our sample membranes. With our present genes identified, we ran a series of analyses to better understand what proteins and pathways are being expressed and what interactions are occurring. We later ran our omics data through the Ingenuity Pathway Analysis database, overlaying our data with publicly available fetal growth-restricted omics data. Distinct expression of membrane proteins present show promise in understanding the role of cytoskeletal development, inflammatory regulation, and cellular signaling in both healthy and adverse pregnancy outcomes. The highly expressed proteins from the previous slide are seen highlighted here in this picture. The numerous lines connecting them to other proteins demonstrate a high level of protein-protein interactions, which could prove useful when determining what protein target sites could be potentially used for delivering drug therapeutics for pregnancy complications. Displayed here are genes from my network overlaid with publicly available fetal growth-restricted data. Highlighted in blue are the present genes involved in insulin growth factor 1 signaling, a signaling pathway that is crucial to healthy fetal growth during pregnancy, meaning that these 2 highlighted genes could potentially be used for drug therapeutic delivery. From my results, we were able to identify a collective network of omics data that is characteristic for placental function under optimal conditions, as well as compare them to fetal growth-restricted data to identify genes that may be useful in determining future drug therapeutics to combat IUGR. I'd like to thank you for listening in to my presentation, as well as extend some gratitude to my lab at the Cincinnati Children's Hospital, the Ronald E. McNair Scholars Program, and the brave women who share their experiences with pregnancy complications to end the stigma surrounding maternal issues.